Hello and welcome to my channel. Today we are going to discuss the two important topics from your class 12th NCERT from Human Health and Disease chapter. That's your cancer and HIV. Each year you can expect minimum of 3 questions from these two topics. So let's discuss it today. Like imagine your cells becoming your enemy or a virus that's slightly destroying your immune system. So today we are covering two of the most important and most asked topics in the NEET biology. That's your cancer and HIV, fine, also called as AIDS. And trust me, after this video you won't need to revise them anywhere else, fine. So let's move into the video, fine. So we will start first with the cancer, fine. Cancer, fine. So like if we further define this cancer, so what's this cancer? So cancer is basically a disease in which the cells divide uncontrollably, fine. So what's now cancer? It's simply a disease where cells grow uncontrollably, fine. So normally if we talk about a normal condition, what happens normally when the cells are like divided, they divide under a control mechanism, fine, which are regulated by the specific genes. But what happens in a cancer, this control is totally lost, and what happens, the cells keep divide and they form a large mass that we call as a tumor. So if we talk about a normal cell, the normal cell, it divides like controlled system. But what happens to a cancer cell? The cancer cell, it keeps dividing like without any proper system, but it keeps dividing and they finally form a mass, and that mass we call as a tumor. So you need to remember it. This is the important, fine. And the mass which is formed by the repeated divisions or uncontrollable divisions of the cells, we call that mass as a tumor, fine. Now let's move into this tumor, fine. Basically this tumor, it has been classified into two main types. First one that we call as a benign tumor, and second we call as a malignant tumor. Fine. So if we talk about this benign tumor and this malignant tumor, they are totally two different things, fine. So let's first move with the benign tumor. So this benign tumor, it's actually a type of a tumor. It's a localizer tumor, fine. Localizer here means that this tumor does not spread, fine. So it's a localizer tumor, and this does not spread. Fine. It means that this very tumor, it's localized to a particular tissue or a particular organ, and it doesn't invade or affect other tissues, fine. So this is a localized tumor. And this malignant tumor, this tumor, it is very dangerous. Why it is dangerous? Because it keeps invading the other tissues, normal tissues, fine. And this malignant tumor, it's actually the true cancer. This benign tumor, they can be small harmless outgrowth also, so not dangerous that much. But this malignant tumor, it's very dangerous. Why? Because it keeps spreading, it keeps the invading other, fine. So this malignant tumor, it possesses a property that we call as, that's important, and it's asked every year in the NEET, that it shows metastasis, fine. So basically it shows metastasis, fine. And what is this metastasis? This metastasis is basically a property that means the spreading property. It's a spreading property. So when I talk about the spreading property, this means that it's shown by this malignant tumor, that's metastasis, okay, through which this malignant tumor, it invades the other normal tissues also, fine. Now let's move into another topic, that is the causes of the cancer, how this cancer is caused, fine. Let's move into the causes of this cancer, fine. So firstly, when we discuss about the causes of cancer, we have to first discuss the carcinogen term. So basically, if we try to define this carcinogen is simply the agent causing cancer. So all the agents, whether it be the physical, it be the chemical, or it be the biological agent, fine, which may induce or which may cause cancer in a cell, we call that as a carcinogen, fine. So basically this carcinogen, also important with respect to the NEET, often asked that what are the agents that cause the cancer, and you need to remember that the agent is carcinogen, fine. Now there are the different types of the carcinogens. If we now talk about these carcinogens, so these are the cancer causing agents, it has got the different types. Now let's move into the types of the carcinogen. If we first, there is a physical carcinogen, fine. So these are, like example we can take as the UV rays. So UV rays, basically these are the physical carcinogens, fine. If a person or if any cell is exposed to UV radiations for a longer period of a time, what will happen? Cancer may like invade that cell, fine, meaning skin cancer is observed, fine. There is another class of this carcinogen that we call as a chemical carcinogens, and there's a big example, that's the nicotine, that's present in the cigarettes or tobacco smoke, fine. So these are some chemical cancer causing agents. Another, there can be viruses or biological carcinogens. So there are some actually viruses which may cause or which may induce cancer in a cell, that we call as a biological carcinogen, these carcinogens, fine. Now another term you need to remember is that oncogens and proto-oncogens. So basically there are the two terms, oncogens and proto-oncogens, fine. Firstly, if we define this proto-oncogen term, this proto-oncogen term, it's simply these are the normal genes, fine. These are the normal genes. fine, that regulate the normal division of the cells, fine. So whenever the mutation, what's mutation? Mutation is changing, fine, changing the genetic setup, okay, of any cell. Whenever the mutation occurs to this proto-oncogen, what happens? Mutation occurs to these proto-oncogens. What happens? They get mutated, and when they get mutated, they become oncogens. And when they form the oncogens, the result is that there will be uncontrollable division, uncontrolled division, and result, there will be cancer. Fine. So what I told there, basically there are the two types of the genes, that's oncogens and proto-oncogens. What are the proto-oncogens? These are the genes that regulate the normal cell division, that are the normal genes. But whenever there occurs mutation by any cause, fine, so these proto-oncogens, these are converted into the oncogens. And these oncogens, what does they do? They cause uncontrollable these cell divisions and finally causing the cancer, fine. Another thing we will be discussing, that's your tumor suppressor genes, fine. Tumor suppressor genes. Example we can take as a P53. This P53, it's a kind of a tumor suppressor gene. This tumor suppressor gene, fine. What does the function it perform? It performs like it stops the cell division. Stops cell division. Also, what does this do? It repairs your DNA. So tumor suppressors are the genes that basically stop the uncontrollable cell division. Also, they repair the DNA, so they don't let the mutation like take place. Fine. Also, they maintain and regulate the cell cycle. But what happens whenever this tumor suppressor genes, these are damaged? This P53, it's damaged. What happens? Cancer develops. क्योंकि इस P53 जीन ने क्या किया था? इसने एक सेल डिवीजन को कंट्रोल में रखा था, ऊपर से ये डीएनए को रिपेयर करता था, म्यूटेशन इसने नहीं होने दिया था. बट व्हाट हैपेंस व्हेनेवर दिस P53 जीन, दिस ट्यूमर सप्रेसर जीन, दिस गेट्स लाइक डैमेज्ड, कैंसर स्टार्ट्स डेवलपिंग. फाइन. So this was about like the causes of the cancer and some terms like oncogens and proto-oncogens, and after that we talked about the tumor suppressor genes. You need to remember that it's P53. Fine. Now moving into another topic, that's the cancer detection. So these are, now we will talk about some methods through which we can detect the cancer in a patient. Fine. So there are some methods. What are those methods? Like first, that's biopsy. That's most important. So if we suspect that any tissue it's suffering from the cancer, ठीक है, कोई टिश्यू है, हमें इस पे शक है that इस टिश्यू को शायद कैंसर हुआ है. So what we perform, we take a small tissue sample, fine, and whenever we take the small tissue sample from that, we study it, okay, and when we study it, we then came to a result that this tissue suffers from the cancer or not, fine. So there are other some tests or some methods through which we can detect the cancer. That's your regulatory blood test. Also imagining, fine, that's your CT scan, MRI, etc. So these are some methods through which we can like detect the cancer. Fine. Also, there's a spatial test. Like pap smear test. Fine. So this pap smear test, what does it like? It has a function, or it perform, or it diagnose. It diagnoses cervical cancer earlier. Fine. So this was about like how we can detect a cancer in any living tissue. Fine. Now there's moving to another topic, that's the treatment for the cancer or cancer treatment. Fine. So basically now we will study that how this cancer can be treated in any tissue if that tissue is suffering from the cancer, like how we can treat it. Fine. So first, that's surgery. It simply means that removing of tumor. Like if any organ or any tissue is suffering from the cancer or it has developed the tumor, we can simply perform a surgery and we can remove that very tumor from that tissue. Fine. Also there's another class of treatment, that's your radiation therapy. Fine. What does this radiation therapy do? It kills the cancer cells. So basically it destroys those cells which are like repeatedly dividing. Fine. Also chemotherapy. So what does we do in this chemotherapy? We just inject some chemicals in a patient suffering from the cancer, and what those chemicals do? They kill those fast dividing cells. Chemicals kill fast dividing cells. Fine. So basically these radiation therapy and the chemotherapy, they have got some side effects. What are those side effects? They cause hair loss. Fine. Because normal cells are also affected. Why affected? They target these chemotherapies, these radiation therapies, they target the cells which are like repeatedly dividing cells. For example, your eyebrows, your hairs, they are repeatedly dividing, although they are the normal cells, but they also got the property of repeatedly dividing. So these chemotherapy and radiation therapy, they don't target only the affected cells. What do? They target all the cells which are repeatedly dividing. Fine. So this was all about this, like how we can treat the cancer. And if we talk about the prevention, how we can prevent the cancer. Now a person is like healthy, how we can prevent cancer in a healthy human. Fine. So first, he should avoid smoking. Why? Because the smoking, it contains the carcinogens that we discussed. What are those carcinogens? The chemical carcinogens, like nicotine is present there. Fine. Also, he should maintain healthy diet. Okay. Also, he should perform regular checkups, like regular blood tests after a particular interval or a fixed interval. Regular checkups. Fine. So this was all about the cancer. Fine. So this was all about the cancer. We first discussed about what is the cancer, then we discussed about the tumor and its types, then we discussed about the carcinogen, types of the carcinogen, then we discussed the oncogens and proto-oncogens. After that, we talked about the tumor suppressor genes. Then we just moved into a topic that was your cancer detection. After we moved into the cancer treatment, and at last we talked about some preventions, that how we can prevent the cancer in a patient. So this was all about the cancer. Fine. So now moving into another topic, that's your HIV. This is an abbreviation. If you look at the full form of this, it stands for this HIV. It stands for Human Immunodeficiency Virus. Fine. So basically, this HIV is a virus which affects the humans. So what does this HIV do? It causes a disease. And that disease we call as AIDS. Also abbreviated form. Its full form is AIDS. It stands for Acquired Immunodeficiency Syndrome. Fine. So, if we talk about the target of this HIV virus, whenever this HIV virus, it enters the body of any person, what is the target site for this HIV? The target sites are helper T cells, another important objective, helper T cells. Fine. And when they target these helper T cells, what is the result from there? What happens? If we move into now the result after targeting these helper T cells, the immune system becomes weak. So as you know that your body, it has got the immune system which protects you from the various these pathogens. Fine. So this immune system, it becomes weak. Fine. And whenever this immune system, it becomes weak, your body is unable to fight infections. Fine. So your body, it becomes unable to fight the infection. So what happens? Multiple pathogens, they attack the body, and your body suffers from various microbial diseases. Fine. Now, if we talk about the structure of this virus, fine. If we talk about the structure of this HIV virus, fine, Now, it comes under the class of retrovirus. So it's a retrovirus firstly. Fine. It's a retrovirus. You need to remember that it's a retrovirus. Fine. So it contains. If we look, what does it contain? It contains RNA and reverse transcriptase enzyme. Fine. So this was all about the structure of this HIV. That what does it contain? It contains RNA as their genetic material. Also, it contains reverse transcriptase enzyme. So now, if we look at the life cycle of this HIV, very important for the NEET. Examination. So what happens? Firstly, this virus, it enters your body. Virus enters your body. Firstly, क्या होता है? ये virus आपकी body में चला जाता है. Then what happens? This virus, it attaches the helper T cells. After entering the body, it attaches to helper T cells. After this attaching to helper T cells, what does this do? RNA gets converted into the DNA. By which enzyme? That's using reverse transcriptase that we talked about here. Using reverse transcriptase. Fine. Then what happens after that? This viral DNA. Now, this viral DNA, it integrates into host DNA. अब ये होस्ट के डीएनए में इनक्रिप्ट हो जाता है. फाइन. होस्ट मतलब नॉर्मल पर्सन, हेल्दी पर्सन जिसमें ये वायरस चला गया था. अब क्या होगा? न्यू वायरस विल बी प्रोड्यूस्ड. इसकी बॉडी में न्यू वायरस विल बी प्रोड्यूस्ड इन दैट पर्सन और इन दैट पेशेंट. फाइन. देन व्हाट हैपेंस? सेल बर्स्ट हो जाएगी, जिसकी वजह से ये वायरस रिलीज हो जाएंगे. ये सेल्स, हेल्पर टी सेल्स, ये बर्स्ट हो जाएंगी, जिसकी वजह से ये वायरस रिलीज हो जाएंगे इसकी बॉडी में. देन व्हाट हैपेंस? ये साइकिल रिपीटेडली चलता है और इसकी वजह से सारी इम्यूनिटी जो है, हेल्पर टी सेल्स, ये सारे डिस्ट्रॉय हो जाते हैं. फाइन. सो ये था इसका लाइफ साइकिल HIV का, कि कैसे ये हेल्पर टी सेल को इन्फेक्ट करता है और फिर पूरी बॉडी को और पूरी इम्यूनिटी को ये डिस्ट्रॉय कर लेता है. फाइन. नाउ सबसे इंपॉर्टेंट अगर हम आते हैं इसके ट्रांसमिशन हाउ दिस वायरस इज ट्रांसमिटेड. फाइन. How this virus gets transmitted. Fine. So basically, if we talk about the main modes of the transmission, इसकी कौन से मेन सोर्स है या मेन वजूहत है जिसकी वजह से एक पेशेंट को, एक पर्सन को ये HIV लग जाता है. फर्स्टली, दैट्स वेरी इंपॉर्टेंट, नीट के लिए भी इंपॉर्टेंट, दैट्स अनप्रोटेक्टेड सेक्सुअल कंटैक्ट. If a male or a female, it's suffering from the HIV and it maintained a sexual intercourse with a healthy individual, अगर उन्हें unprotected sex किया, that it means that they did without using the protection or the condoms, what will happen? This virus will infect the healthy one also. It's fine. So this is the first cause. Second is using the infected blood transfusions, किसी इंसान को हमने किसी सिरिंज से या किसी इंसान को हमने खून चढ़ाया किसी दूसरे इंसान का, बट जिसका उसको खून चढ़ाया वो HIV इन्फेक्टेड था. जो वो खून उसकी बॉडी में, एक हेल्दी इंसान की बॉडी में चला जाएगा, ही विल आल्सो बिकम इन्फेक्टेड विद दिस HIV. फाइन. देन आल्सो अनदर कॉज इज शेयर्ड नीडल्स. लाइक किसी पेशेंट को हमने इंजेक्शन किया एंड वही सेम इंजेक्शन वी टुक एंड वी इंजेक्टेड और वी परफॉर्म द इंजेक्शन टू एन अनदर पर्सन. अब ये पॉसिबिलिटी हो सकती है जो कि पहले वाला इंसान है, उसको हमने विद द हेल्प ऑफ द सिरिंज ब्लड निकाला था या कुछ इंजेक्ट किया था, उसको HIV था, वो HIV लग के उस सिरिंज से दूसरे इंसान के में चला गया जो कि हेल्दी था, जिसमें HIV नहीं था. एज अ रिजल्ट वो पर्सन भी इफेक्ट होता है HIV की वजह से. ऑल्सो अनदर कॉजेज, अनदर मोड ऑफ ट्रांसमिशन फ्रॉम इन्फेक्टेड मदर. ठीक है, ये चाइल्ड में जा सकती है ड्यूरिंग हर प्रेगनेंसी. अगर मदर इन्फेक्टेड HIV से, जो बच्चा उसकी पेट में होगा था उसको भी HIV लग सकता है. फाइन. अब कुछ यहाँ पे ये है लाइक गलत मिसकंसेप्शन है लोगों को कि शायद HIV हैंड शेक करने से या खाना शेयर करने से या हग करने से या यूटेंसिल्स दूसरे की यूज़ करने से भी HIV लग सकता है. बट HIV का वो वैसे ट्रांसमिट नहीं होता है. फाइन. ओनली मोड ऑफ ट्रांसमिशन आर दोज विच आई डिस्कस्ड हियर. फाइन. अगर हम किसी HIV पेशेंट से लाइक हैंड शेक करें, उसको टच करें, उसके बर्तन में हम खाना खाएं, उसकी वजह से HIV ट्रांसमिट नहीं होता है. फाइन. सो दिस वाज अबाउट. सो यू नीड रिमेंबर दैट व्हाट आर द मोड्स ऑफ द ट्रांसमिशन एंड व्हाट आर नॉट. दिस मे कम इन assertion and reason type questions also, that by hugging or by handshaking or using the utensils or articles of a infected person क्या HIV लग सकता नहीं लग सकता है. सिर्फ इन मोड्स की वजह से HIV ट्रांसमिट हो सकता है. फाइन. Now if we look at the symptoms of this AIDS, अगर आप एक इंसान को HIV ने इन्फेक्ट किया, अब उसमें AIDS डिजीज होगी. Now what will happen? This AIDS हो गया. What are now the symptoms of this AIDS? Fine. उसमें क्या होगा? फीवर आएगा. The person will or the patient will suffer with the fever. उसमें weight loss होगा. उसमें chronic infections आ जाएंगे because body is no longer now able to like fight with the diseases or the infections. उसमें chronic infection और उसकी immunity weak पड़ जाएगी. Immunity will weaken. Fine. Now if we talk about how we can diagnose this HIV, diagnosis important also. Diagnosis of this HIV. Fine. So there's a test that we call as ELISA test. ELISA. Very important. 1000 बार नीट में पूछा गया है. Fine. What's this ELISA test? It's used for the detection of HIV. Fine. ये क्या करता है? Detect antibodies को. Antibodies को. Fine. Now, so if we talk about now the treatment, if now a person suffers from the AIDS, what will be the treatment? Right now there's no complete cure for it. No. अभी तक कोई कंप्लीट क्योर नहीं मिला एड्स का, बट इट्स कंट्रोल इज पॉसिबल बाय यूजिंग ART, दैट इज एंटी रेट्रोवायरल थेरेपी. इसकी वजह से वी कैन लाइक increase the lifespan of a person who is suffering from the AIDS, but we can't cure him this completely. So this ART therapy, it's induced to a person suffering from the AIDS. It can't completely cure it from that person from the AIDS, but what it can do, it can like enhance the lifespan of that person. Right? So now how we can prevent this AIDS? If we talk about this prevention, so there are some prevention that we can like follow to to be safe from this HIV. Fine. Like firstly, have a safe sex. Means that whenever you are having a sexual contact, the person use the protection system. Second, use sterile needles. Also there should be awareness. Fine. So if you understood now this properly, you have like covered almost 100% of the NEET questions from the cancer and HIV. Fine. So comment done if you completed this, and I will bring more high yield videos like this. Fine. So this was all about it. Thank you for watching it.